Good morning. Yeah, I got few emails that the lecture has been planned at 11 o'clock, your university lectures. So once again, we'll be having very few friends with us at this point of time, but no worries. So today we'll talk about skeletal muscles. Quite conceptual. The reason behind it is that we have taught part of it, right? We have taught part of it, and we'll be adding something extra. This will be helpful for us, right? So here is something which we have discussed in detail, right? This is skeletal muscle structure, right? And this is what we are watching is sarcomere. So in this, we talked about that there are two types of bands, right? Way back when we said that uh, it is like actin, thin, myosin, muscular, thick, right? On the myosin, there'll be the cross bridges. So today we'll see the cross bridges, how they work. Then these were like Z lines, right? So in between two Z lines, so that is what it is, right? And this Z line was also called as the that Zwischen Scheibe, right? It was Zwischen Scheibe, right? Basically, it is what? It is a German word, and German word for that is the intermediate disc, correct? It is the intermediate disc. Then regarding these bands, that is I band and A band, right? What exactly were they? Well, we remember that this one, actin, right? This is thin, and myosin, that is thick, right? Actin is thin filament, and the myosin, they are the thick filaments. And when we talk about them, the most important point is that about their characteristic, right? Actin is thin and shiny because we said that it is isotropic, right? It is isotropic, and when we say isotropic, that means it has got one refractive index. So because of one refractive index, it would be I, right? That is isotropic, so it is I. So here it is our I, right? Isotropic. And when we say isotropic, that means this particular band is where there is only actin, right? So that's where the actin is, right? So here if you see this portion to this portion, right? This portion, this one, right? This is only actin, right? And A band where there is myosin, right? This is only the myosin. And why we call it A band? Because it is N isotropic, right? It is N isotropic, right? We have talked about it. N isotropic, that means it has got more than one refractive index, right? N isotropic, more than one refractive index. Refractive index. M line is the middle line, right? M is for middle, so that is not a big problem. M line is the middle line. Thin filament, thick filament, myosin, actin, all good. These two new gentlemen, tropomyosin and troponin complex. That is what we'll be studying today. And then we'll be proceeding for our some other interesting facts, right? That is the clinical aspect. So with this, yeah, today because there are images, so I have changed the background, but I think black was better. Anyway, no problems. So here it is, the muscle proteins, right? Muscle proteins. So we'll be writing it in the form of brain map that there are four types of muscle protein, four types, right? One, what is called as the contractile protein. And these contractile protein, right? So contractile protein, actin and myosin, right? Actin is thin, delicate, and myosin is muscular, thick, right? So actin, myosin, and then there will be cross bridges. We'll come to that. So this is contractile. Then there would be some supportive protein, right? There would be supportive proteins. And in these supportive protein, we need to know about one, dystrophin, right? It is the dystrophin, which will be our supportive protein. Then another important protein that would be the desmin, right? Desmin is important, and some spring-like structure, it is called as titine. Putting a mark, right? These are important. The third type of protein, which is called as the protein which is meant for relaxation. So it is a relaxation protein, right? It is the relaxation protein, and it is called as the Circa, right? Circa. Circa stands for smooth endoplasmic reticulum, right? So that is easy. C is for calcium, and A is for ATPase. Now we know that this smooth endoplasmic reticulum, that is like a storehouse, right? So basically, Circa is a calcium activated pump. So it's a pump, right? It is this ATPase, right? Which will be, so this will be an active pump. And the fourth one, it is regulatory. Regulatory proteins mean what? Regulatory protein, they will be controlling it, right? Whether the muscle should contract or it should not contract. So for that, those two new characters, that is the tropomyosin, right? Tropomyosin and the troponin C. So these two, yeah, and troponin C. So these two, they will be the regulatory protein. These are the proteins. This is what is in our dish for today, right? Now see, actin, myosin we have already talked about, right? So let's start with the supportive proteins. So in supportive proteins, one, dystrophin, right? So this dystrophin protein, it's a rod shaped, right? It's a rod shaped, and basically it connects cytoskeletal of muscle with extracellular matrix, right? So think it like that, the muscle and the extracellular matrix. So something which is outside that cell, extracellular matrix, and this is the muscle, right? So it connects cytoskeleton of muscle with the extracellular matrix. So due to this, that muscle tissue, right, that muscle tissue remains in its proper state. So this whole complex, right, both of them considering together, that is called as costamere. Sarcomere was what was between the Z lines, and costamere is when this particular sarcomere is connected to extracellular matrix, and that is called as the costamere, right? Costamere complex, right? So here it is, say, sort of this, right? Here it is the costamere structure. So we have got extracellular matrix and our muscle cell, it is connected with, so that is dystrophin. This is where the dystrophin would be used, right? So this is our costamere. So costamere, and between Z lines is the sarcomere. Okay. Apart from that, one common question which you must know, and it would be a big clinical thing, right? This dystrophin, putting a mark, right? When there is mutation of dystrophin, when there is mutation, it leads to something which is pretty serious, right? This is what is called as the muscular dystrophy, right? It is called as the muscular dystrophy. but there is a name which is given to it. It is called as the Duchenne muscular dystrophy, right? It is called as the Duchenne muscular dystrophy, right? And many questions they can be asked from this. But do remember, it is dystrophin mutation. As we'll proceed, yes, we'll be talking in detail about this particular condition. Right? So this is first. The second one was Desmond, right? Second one was Desmond. And for Desmond, this is also one of the protein, right? And it belongs to what is called as the intermediate protein or the intermediate filaments. So it is into those intermediate filaments. But there is one another important use. Put a mark. It is considered as tumor marker. It is a tumor marker for sarcoma, right? For sarcoma. So what exactly is tumor marker? That means this Desmond can be utilized to detect the presence of sarcoma, whether there is any presence of it or not. How to do it? Well, that is not we'll be discussing right now, because all those procedures will be taking separately in the form of tumor marker. One complete session will be on tumor markers. Which are the tumor markers that can be utilized? But do remember that when we talk about Desmond, tumor marker. Out of all these protein, the third protein, and this is the protein which is very vital, and it is what is called as the titine. Titine, right? Just to remind you, we are talking about the first group, that is the supportive protein, right? We are talking about supportive proteins. So first was dystrophin, second Desmond, and third is titine. And this titine is a spring-like protein, right? So this titine is also called as the muscle spring. Muscle spring. So what's the function of spring? Spring gives elasticity. Elasticity. When we say elasticity, it means that we stretch something, and when we release it will come back to its original shape, right? That is elasticity. This is what we get with the titine, right? So it is like even if the muscle has stretched, muscle stretches, right? Muscle stretches, but then it comes back to original shape and size. It won't remain into that state. So this is because of titine. Now, interestingly, you'll find that titine is, this is present, right? Titine is present in skeletal muscles and the cardiac muscles, right? It is present in skeletal muscle and cardiac muscle. It is not present in smooth muscles, right? So big absent in smooth muscles. Do remember star, right? Whenever I'm putting star, you know that question has appeared somewhere in the competitive exam. So it also works in muscle relaxation. Means you are talking about titine. Yeah, definitely, right? It would work. So that's why this titine is why it is so important. See, I'll show you one condition. Okay, see here, right? I have put few images randomly, right? So today it might happen like that, but say over here, this is where I wanted to talk about, say, titine, right? When there is mutation, right, in cardiac muscle, right, in cardiac muscle, whenever there is titine mutation, that means titine will be losing its characteristic. That means its spring-like action mechanism is lost. So that will lead to what's called as the dilated cardiomyopathy. See the thing. heart is dilating, but then it is failing to come back to its original shape, original position. Right? This is like a normal heart, right? And this one is dilated cardiomyopathy. So today you'll come across several such words. Just keep them in mind and try to associate. Right? What I mean to say is try to associate those keywords which we are discussing. So we talked about, right, this costamere, that was a new word for today. Dystrophin, Desmond, and Titine, right? These are the three important supportive features, right, or the supportive proteins, right? Yeah, it loses elasticity. Exactly, it loses elasticity, and elasticity means coming back to original shape and the size, right? That is the specialty of Titine. Okay, on to our next one. Okay, now these are called as the regulatory protein, right? These are, so you might have noticed that we have skipped Circa. Yeah, deliberately we have skipped Circa because once we'll understand the entire contraction process, then during the relaxation process, we'll see that from where this Circa's importance arrives. So this regulatory protein, for this we have got two, right? Two. One is tropomyosin, right? It is the tropomyosin. That is one. And the second is troponin C, right? Troponin C. Now, before we start, one important thing, that skeletal muscle contraction is voluntary, right? So, our skeletal muscle contraction is voluntary. So, that means when you are at rest, during that point, there is no contraction due to these regulatory proteins. It means that those actin myosin filaments, they are constantly interested that yes, we should interact, right? We should talk, and then we should have this contraction phase. But it is the tropomyosin and troponin C, right? They are regulatory proteins, so they are keeping the things into relaxed way, right? So they are not allowing actin myosin to create a session. Fine. So here it is how it is really formed. See, all these, this is a combination, right? So you can say all these individual, they are all globulin proteins, right? Actin, right? Actins. So there is tropomyosin and troponin complex. So what it is happening is that when the myosin, that muscular myosin will come, and it would try to come and bind at these sites, these black sites, right? These black sites. But all those black sites, they are covered, right? All those black sites, they are covered because it is the tropomyosin which has overlapped that entire area, right? So it is tropomyosin binding site. Tropomyosin has covered the myosin binding site, right? So we can add over here that it is the myosin binding site, because over there the cross bridges from myosin, they will come, they will attach, and then they will give a power pull, right? And then the muscle would keep on contracting, right? But this myosin binding site, right, it is covered. And covered by whom? Covered by tropomyosin, right? Covered by tropomyosin. So he is like that villain, right? He is over there. When the calcium comes, now he is greedy. When the calcium comes, so that calcium is bound to troponin, right? Calcium is bound to troponin. So this is step number one. And this troponin, right, will pull, right? It will pull, it pulls tropomyosin. So when this happens, myosin binding site that is exposed, and the process continues. Now, the myosin cross bridges, they can come and they can bind at this point, right? So what we understood is that when the calcium comes, troponin, it binds and then pulls the tropomyosin, right? And now actin myosin, they can meet. So but this is the basic. So when the actin myosin contraction starts, then things they are into action. So see here some graphical images, right? Some graphical images. So that's the actin and this one is myosin. And see these are the heads which will be going and will be connecting to actin, right? So these are the myosin heads and all those black dots in this case they are yellow, right? In this case they are yellow right now over here. All these yellow dots, if you see, they are all covered, correct? They are all covered. Well, right now muscle is in resting phase, right? This is resting. Now in presence of calcium, right? In presence of calcium, things would change. So this is troponin, then tropomyosin, right? Troponin, they are like pegs. They are in standing in between, like those small pillars. And this tropomyosin is like a rope, right? It is like a continuous rope. And that rope, it is actually covering all the connecting sites. So there is no way to do the picnic, right? And troponin, these are like pillars, right? In between, they are like pillars. And obviously, when none of the site is exposed, so bichara myosin is alone. At this point of time, myosin is alone. Now here comes the calcium, right? So when the calcium arrives, over here calcium was already on the way, right? It was en route. This is calcium, right? This one is calcium. So those calciums, they are now coming into picture. So as that calcium arrives, right, it attaches, right? When it attaches to that troponin, and that troponin, it then pulls the tropomyosin, right? So that's what we saw. It pulls the tropomyosin. As soon as the tropomyosin is pulled, now see the sites, all these yellow sites, see they all are exposed, right? All of them, they are exposed. So this is where the head will connect and that is exposed. Still there is some other mechanism and we'll be learning that. Right, so these binding sites, all binding sites are open, right? So these are the binding sites. They are open or exposed, right? Obviously myosin binding sites. Myosin binding sites, they are open and the action starts, right? And the action starts. Good one. And see how they move, right? And they'll keep on moving till the ADP and the phosphate is there and then it is released, and when the ATP comes, once again it will come back to normal, right? Now we see that, but this is how the entire contraction moves. This is actually what is called as the power stroke. It is called as the power stroke. So today we'll also be watching type of muscles, that what happens when you really want to develop the muscle, what do you want to develop, right? You'll say I want to develop the power. So there are specific muscles for the power and there are specific muscles for the endurance, right? Types, type 1, type 2, type 3. So we'll see that. But all of them, they will be working within the same principle, right? So that's what we understood now, that muscle contraction, it occurs only in presence of calcium, right? So this is the muscle contraction, right? So muscle contraction possible if calcium is present. And this theory has been given a name, right, in which there is actin myosin interaction, and it is called as the sliding filament theory. In sliding filament theory, we'll also watch that when the muscles they really contract, at what point there is the maximum tension, right, maximum power. And it's but obvious that this is myosin, muscular myosin, and this is actin. So when there would be maximum overlap, there would be maximum tension, because then the maximum number of cross bridges from myosin, they are connected to actin. So it would be the tightest possible bond, right? So that is when there is maximum overlap. When there would be maximum overlap between actin and myosin, the strength would be highest. We'll see a very good graph of it, right? Chalo, so let's understand the sliding system or the sliding theory, right? Just the name, sliding filament theory. Theory is just a name, otherwise so everything is sort of practical, right? So here are the stages. So in the stage 1, right, these are the stages. Now this is stage 1. This is at rest, right? So that's from where we start. Here, the ATP, it comes and binds, right? So at rest, ATP is there. Now it is sitting and ATP comes. Because things are at rest, the myosin head, right? So we'll call this as myosin head, right? That's the myosin head. It is at 45 degree, right? Myosin head is at 45 degree. ATP comes, and now the ATPase enzyme is activated, right? It is activated. Still remember, we are into resting phase, right? Nothing more has occurred over here. So when this ATP comes, so from ATP, it would be broken down to ADP plus the phosphate bond. Right? And that is by ATPase. So far easy. So here it is broken into, right, ADP and the phosphate. So that's the resting state. Now when it breaks, it comes out from the resting state, right? Because now the myosin head, right? Myosin head from 45 degrees, it moves to 90 degrees. It moves to 90 degrees. So here it is, if we draw, so this is 90 degrees, right? And over here, we can say that this was like 45 degree. I'm drawing this thing over so that when you revise, you don't have to even read the contacts, right? Straight away, you just see the figure and you can move further. And another is ADP and the phosphate bond, right? ATP is broken. In other words, ATP is broken. On to the next one. Meantime, the calcium has already acted upon binding sites, right? Calcium. So we have already talked about it. So the calcium has already created good environment, right? So created exposure, right? Exposure of binding sites, correct? Binding sites. So that's it. Now there is no one to stop actin and actin myosin interaction starts. Right? So actin myosin interaction starts. So there might be some music over here, right? Right? Some music. The interaction has started. This is also called as cross bridge formation. Cross bridge formation, right? Because this is the first touch. Fine. So cross bridge formation. Now, because of the presence of ADP and phosphate, this is the most interesting part. In fact, once I'll explain this entire thing, you have to close your eyes and you have to visualize, right? Yeah, that middle fibers, right? Those middle fibers, they are the heads, right? So they are the heads. And what we are watching is for one head. This thing happens with all the heads, but right now just think about one head, right? Because names are very interesting, right? Seriously, names are very interesting. So, so now think it like the head is there, and this head remains in good mood till ADP plus phosphate is there, right? And it happens like then as it connects with the actin, and it really pulls, right? And that is what is called as the power stroke, right? So from 90 degree, right? It was at 90 degree, it it pulls. So actually this becomes now 135 degree, right? So it is his pull, right? It pulls. But what has happened? See, it was attached at this point. At this point. Over here, it was attached at this point, right? So after attachment, it is pulling. So the whole this thin fiber is moving, right? The whole thin fiber is moving. The whole filament is sliding, right? And that's why this thing is called as the power stroke. It is called as the power stroke, right? So that ADP plus phosphate, till they are there, right? It will keep on pulling, right? Now ADP and phosphate, they are released, right? So ADP plus phosphate, they are released at the peak of power stroke, right? It is released, and this myosin head, now it is waiting, right? It will stop going for further power stroke. So myosin head waits, right? Waits for whom? Waits for new ATP, right? So waits for new ATP, right? And when the ATP comes, and then ATP attaches, so this is the ATP arrives, right? Here is the ATP arrives, and ATP attaches, arrives and attaches, and this ATP will say, okay, beta, shant, right? Come back to normal, right? So myosin head would say, okay, right? So this myosin head returns, right? Back to original state. Myosin head back to original state. Now this is happening with all the heads, right? That's why we'll be requiring large number of ATP, right? So back to original. But over here, again, as soon as ATP comes, so then this is the ATP is, it comes into picture, right? And as it is broken down to ADP plus phosphate, so then that is the so stimulatory for head. He forgets about the advice of ATP, ke beta come to normal. He said, "Arey, forget." And once again, right, he'll say, ke, okay, connected, right, from 45 degree, right, this is 45 degree, then from 45 degree, it connects, and then it gives another and that is power stroke, right? Power stroke, right? And then this ADP and phosphate, right, they are released. So this waits for the next ATP. When ATP comes, so he tells, okay, okay, beta, back to normal. So he'll come back to normal, right? Back to normal. And the process keeps on repeating itself, right? So this ATP comes, right? And he returns, right? And then he returns. This is the entire process, how it really happens, right? This process keeps on getting repeated. Now think about in this, the way we looked. So here is that actin, and here is all those heads of myosin. So when the ATPs they come, so it does not go back to its until ATP comes. No, it is the ATP's advice, right? कि चलो बेटा go back to normal, so he goes back to normal. Otherwise, he'll remain into that position. So that means that ATP is needed for relaxation. Yes, it is needed, and this is the reason that during action, right, during rigor mortis, right, or when the people die in action on the front, during action, during firing, right, and suddenly the headshot and then they out. So during that contraction phase, now there is no time for relaxation, right? So they remain into that stiff position. So ATP is what takes the head back to its original state, right? And then ATP breaks into ADP plus phosphate, and then comes the that power pull. So all those heads, right? All those heads, they are literally working together, right? Working together, and it is like myosin will remain over there. Only its heads are moving, but it is the actin filament which is sliding, right? Which is sliding. So this is myosin, right? And the actin contracts, 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 contracts. And then when there is maximum overlap between myosin and the actin, when there is maximum overlap, so that maximum number of heads they are connected and they are giving the power stroke, that is where the maximum power would be generated, right? What is the importance of this entire system? Why we understood it? Because when I say that being a medico, you should be very fit. So sometimes when we do the exercise, right? When we do the exercise, so if you take the dumbbells and then dub 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 dub, you start doing it, right? You'll find correct muscles are not developing or power is not developing. Power will really develop when you are training your maximum overlapping of myosin and actin, because that would be when the maximum contraction would be there, right? So you give time, you give time. So that's the reason you just do a simple thing. You take a dumbbell, right, which you find that is, yes, this is comfortable. So when you flex, right, so you say 1, 2, 3, 4, 5. Now give the double time to return. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Bus. Follow this principle and see what happens. Because in that case, you are handling all the situations whatsoever we discussed. All of them. In hypertrophy of muscles, well, in hypertrophy of muscles, in number of actin and myosin filaments, so they remain same, right? They remain same. It is not so that their numbers would increase. But in case of hypertrophy, right, as you said, the size would become bigger, right? The size would increase. So these muscles, now the heads, more and more number of heads they will be involved, because for the muscle, it is not so that all the heads they are involved together, right? Because at times there are not sufficient ATPs to involve all the heads, right? So as you practice, as there is hypertrophy, more and more participants of actin and myosin, they will be involved, right? So that leads to improvement in the power. So in other words, the thing what is called as the cross ridge cycling, it is what is called as the, what your answer is, it is like, correct? Why it is disappearing? Don't know. Let's check it out. Is it happening here also? No. Here it stays. Fine. So that is what is called as the cross bridge cycling. Cross bridge cycling. And that is, in other words, nothing but muscle contraction. Now, what we understood was the basic phenomena, right? Now we'll be understanding it from two different perspectives. First, from the calcium ion perspective. Calcium ion, right? Calcium ion, when it is present, it will lead to contraction. Correct? It will lead to contraction. And when it is absent, it will lead to relaxation. Right? It is leading to relaxation. For the calcium ions, when we go into still more depth, right, that is the calcium channels. There are 3 calcium channels. There would be 2 channels which are for contraction, right? Which are for contraction. And there would be one channel. There is one channel which will be playing its role during relaxation. So for relaxation, there is a separate channel, right? For the contraction, we divide it into 2 parts. One, what is called as the DHPR, dihydropyridine, right? This is a receptor. So it is dihydropyridine, and this is receptor. So as always, we write RS like receptors, right? And this second one is called as the rhinodine. Putting a star. Rhinodine receptor. Now both these receptors, they are having very specific purpose in the form of that this dihydropyridine receptor. When you give calcium channel blockers, right, it will be affecting here. So there will be few drugs like, say, calcium channel blockers, right? These are the class of drugs, calcium channel blockers. So for example, nifedipine, right? Say this nifedipine is a calcium channel blocker. So what it blocks is DHPR, right? It is the DHPR which is blocked. Rhinodine receptors, they are when overstimulated, right? That is when they are overstimulated. So many conditions, right? Say for example, hyperthermia, right? When the temperature shoots, temperature is the fever, right? Hyperthermia is like a fever. But when we say malignant hyperthermia, that means it is uncontrollable, right? When it has gone extremely high. And this is the more heat is generated due to excessive contraction, right? Excessive contraction over here. That would be the keyword, excessive contraction. So in that case, it's treatment. What you have to do something? Obviously, rhinodine receptor blocker, right? Rhinodine receptor blocker. So rhinodine receptor blocker. because these are the classical thing. There is excessive contraction, and when there is excessive contraction, so that is because of ryanodine complex. So here the treatment would be something which blocks. So say for example dantrolene sodium, right? So this is dantrolene sodium. Yes, it is not necessary to remember all the names at this point of time. It is completely fine if you forget it, right? But every time you will see these notes, right, these names will keep on coming in front of you. And when you'll be actually learning the pharmacology, at that point instantly you will say, aha, this is where these receptors are working. Because that is when these receptors will be coming into so much of detail, so that and all the slots will start fitting into its correct place, right? So that is one. So we have talked about, say, these proteins, right? So we talked about actin myosin, we talked about those supportive proteins, and we also talked about those regulating proteins, regulatory proteins, correct? So we were here, right? Contractile proteins, supportive proteins in the form of dystrophin, Desmond, and Titine. Titine, which was like a spring, right? Which was like a spring, and if this spring is lost, right, that leads to muscular dystrophy, especially like in heart, because they are present in skeletal muscle and heart only, right? And then we talked about regulatory proteins, that is tropomyosin and troponin. This troponin, how smartly in presence of calcium creates a bond, and it pulls the tropomyosin, and when it pulls the tropomyosin, myosin sites, right, those myosin binding sites, they are exposed, and those heads, they come, and in the presence of ATP, it gives the power stroke, right? And when after some time that calcium is released, so once again that tropomyosin will come, and it will come into its original state, and the muscle is relaxed, right? When the muscle is relaxed, it gets more time, so that more calcium comes. When the more calcium comes, once again the whole cycle starts, right? Relaxation, this is the fourth, the final part, and that is Circa, right? So that is the relaxation protein. That's what we'll now talk about. All right, all these states. Oh, they appeared after some time. Huh. So these are now relaxation proteins. ठीक? And it is Circa. Yeah, Circa sounds like, yeah, move aside. So this is smooth endoplasmic reticulum, calcium ATPase. In fact, Siddhant, same thought came in my mind, and at that time some Govinda's movie was going on. Some I don't know, some Sadasa movie, but there was some song also like this. Okay, so this is smooth endoplasmic reticulum. ठीक? This smooth endoplasmic reticulum, it stores calcium. This calcium is released. When this calcium is released, so here is what? Here is our ryanodine receptor, right? Here is our ryanodine receptor. So this receptor, right, will say, okay, calcium, go out. And this calcium will now take part into muscular contraction, right? Muscular contraction by connecting to troponin C, and then that troponin C will pull the tropomyosin and the whole game starts. Then muscle contraction is complete. This calcium is released, right? This calcium is released. This is pumped back by what's called as the Circa, right? So this is the meaning. This Circa, smooth endoplasmic reticulum calcium ATPase, will put this calcium back into the system, right? So this is pumped back. So it is the ryanodine receptor which will take the calcium out and will be available for the muscular contraction, while this Circa will pump it back, right? So that is the function of Circa. And when the calcium is released, so obviously this leads to muscle relaxation, right? It leads to muscle relaxation. Now, as I was talking about that, yeah, this one. That when we say that properties, right? In case of skeletal muscle, it stretches when the length changes, right? And when it relaxes, it once again goes back to its original state. So there is elasticity, and that is because of titine, right? So in the properties of skeletal muscle, we can say one, the property is contraction, and second, the property is elasticity. Elasticity. Right. And when there is maximum overlap of actin and myosin, right? Maximum overlap of actin and myosin. It is the maximum generation of force or tension that is highest. But that's the logic. It is the overlap, right? It is the overlap of these two which generates maximum force or tension, because there would be so many heads connected to each other, right? So it is so difficult to separate them, so it generates maximum strength, right? So here is, now we watch this. So this particular graph, yeah, this particular graph, right, which is called as the length tension relationship. Everything will boil around actin myosin, right? Obviously. And it is worth visualizing, right? So many things. This color is okay. It is okay. So let's start. During the initial stage, there would be minimal overlapping of actin and myosin, right? Because it is relaxed, right? So there will be minimal overlapping of actin and myosin. So there is less tension, right? Because muscle is relaxed, correct? And now the overlapping starts. So here is now the overlapping starts because of power stroke, right? Because of power stroke, now the overlapping is starting. When the overlapping is optimal, what exactly we understand by optimal? Zoom to this thing, this portion, right? This, and we have like this, right? So those actin filaments, when they come near each other, right, that is when the muscle is contracting. But say for the myosin, the power stroke, right, power stroke area is this. It is not along the whole part. So in between portion is not having any of the myosin heads, right? That M line, that central line, that middle line, right? Because that is just the connecting. So obviously, when this active area meets the maximum active area, right? When active area meets the maximum active area, there is the maximum tension. This is where the maximum power is there. This is what is called as the optimal overlapping. Overlapping. Optimal overlapping. If we go back to our that image, right? So I think we should be able to see something. True. See, for time being, I'm just erasing this. See here, there are no heads. So consider that this is the end of the myosin filament. Myosin filament. This is the end, right? And this one is our thin, beautiful actin filament. So any actin filament going beyond this, right? It is of no use, right? So for getting back calcium to cell, is ATP needed? No, no. Right. So this, when this is optimal, right? When this, there is optimal mapping, optimal overlapping, this is when there would be the maximum pull, right? This would be the maximum pull. So that is what I mean by active area. ठीक? And that was the myosin head. That was the myosin head. Then once again, as the procedure continues, right? So myosin acting, they will start going far. Yeah, right. At the peak, this is the maximum power, and once again they start relaxing, and gradually, say it decreases so much so that when it is completely relaxed, right over here, there is no actin myosin overlap. There is no overlap, right? So that is when the tension is least. So it is the least tension. And the cycle repeats, right? Yes, this one is when we have got, it's a different type of muscle fibers, right? This is types of muscle fibers. So type 1, they are like slow twitch, but they are extremely important for long distance. Fast twitch, oxidative, right? That is 400 meters to 800 meters. And for fast twitch, glycolytic, they are for high speed sprints, but they are short, right? 100-meter dash. So that is for short sprints. That's the reason whenever you are, see the fatigue rate, right? You notice the fatigue rate low. That means for the long distance, it is you have to use slow twitch fibers, right? And for the sprints, it is the high twitch, this fast twitch fibers. But their rate of fatigue is very high. That's the reason that when you are planning to run for very long distance, right? If you try to go for sprint, right, immediately, right, these muscles will be tired and person will go into the state of fatigue. Then these group of muscles, they are of no use, right? They won't be able to work. There are some other very classical things about it. For type 1 fibers, right, these fibers, they are small diameter. Their diameter is small, right? These are the small diameter muscles. Red color, right? Their color would be red. Why they should be of red color? Because of the presence of myoglobin, right? So presence of myoglobin. For type 2, here also it is exactly the same. So that's why I'm just putting tick over here. That is presence of myoglobin. For high speed fibers, right, you won't be finding that. So there is no myoglobin. This is one major difference. Because there is no myoglobin, that means they will be looking white. So they are white, right? So don't forget this difference. Red color is because of the presence of myoglobin, which is present in type 1 and type 2A, and in type 2B, you have got white color appearance because there is no myoglobin. Type 1 is, you can say, small diameter, while type 2 and type 2A and 2B, they both are large, right? Large diameter. They are big muscles, large diameter, or the large diameter. Second important thing, as it is working slow, right? So this is aerobic. This is aerobic. Aerobic means in presence of oxygen, right? So here you are getting ATP. ATP would be needed, right? So ATP with presence of oxygen. So oxygen, this would be aerobic. Even over here also, right? It is aerobic, right? So here also like this, ATP is there, right? Plus in the presence of oxygen. But while in case of these sprints, it is called as anaerobic. Anaerobic. As they are consuming ATP so fast, right? So there is practically no oxygen, right? And here you will be getting it by oxidative process, right? Because oxygen is present. While over here, in case of anaerobic, right? Yesterday, do you remember? We talked about glycolysis, right? Glycolysis, when the glucose is broken down into pyruvate, right? So what pyruvate was, right? Pyruvate is 3 carbon, right? This is 6 carbon. So 2 pyruvates, that pyruvate would be broken into lactic acid, right? And it is the accumulation of that lactic acid that gives fatigue. But active lactic acid is as such very good. Lactic acid, it stimulates growth hormone. So that is what gives your muscle the good power. That's why in marathon practice, right, you develop your strength by running long distance. Yeah, hence more lactic acid accumulation. That's right. You develop your strength by running for a longer distance. But in between phase, you go for all round full sprint, high speed sprint, because you want to train all of your muscles, right? All of your muscles, and this is what gives power. And so this is power. This is power, and this one is endurance. Endurance, right? Both are needed. So during your run, we say that's fine, that it is for steady run, right? It is needed. Over here we say this is for short run, right? And this is for all round sprint. But when we'll be understanding the muscles of the leg, you'll find that it is not only for steady run. The way you stand, the way you walk and your posture, it depends on this group. Because when you are standing is like when those type 1 fibers they are involved, right? So that's why it is necessary to train yourself. Now we talk about one more concept, the last one for today, the thing what is called as the muscle twitch, right? So what muscle twitch is? So this is the muscle, right? And we give a stimulus, right? We give a stimulus. We give a stimulus. What would happen? Muscle would contract and then it would relax. Gluteus maximus, yeah. Even medius, right? Medius, minimus, all of them, they are taking part, especially when you will be learning that region, that why it is said that lunges they are more important or the squats they are more important, because they actually train your those muscles. And those muscles, they are as such very important for so many important intimate functions, right? Very powerful, they are very powerfully they are involved, including the core muscles. I don't know, not from your batch, but from previous batch, someone asked this question, that that how can you discuss these things in public? I said, why? It's not part of the medical system, right? Because in abdomen and in lower limb, yes, we have talked so many things about it. But yeah, I think I was blocked over here for discussing that, and then the rest of the session we had to take on Zoom. That was very interesting. When I still, that lecture is has been banned for using such words as if some crime has been committed. Anyway, so this is the stimulus, right? And the muscle contracts. So this is the contraction, and then muscle relaxes. So this is the relaxation. So far, so good. Nothing, everything is fine. Now you keep on giving the stimulus again, again, and again, 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 again. So you give one stimulus, second stimulus, third stimulus, fourth stimulus, fifth stimulus. So muscle will also behave like this: contract, relax, contract, relax, contract, relax, contract, relax, right? That's how contracts is, relaxes, contracts, relax, and it goes on. This thing is called as the incomplete tetanus, right? Remember, you are not allowing the muscle to relax fully. Yes, right? Incomplete tetanus or incomplete titani. Exactly, right? You are not allowing the muscle to relax fully. And before that, you are just putting second one. When you start giving the stimulus so rapidly, right? So once the muscle contracts, you are not at all allowing it to relax. So its graph would go something like this. Its graph would go something like this, right? This is what is called as the complete titani or titanus, right? In this, there is no relaxation, right? Keep this thing in mind, that there is no relaxation, and there is sustained state of contraction, right? So sustained contraction. What's important is that at what rate are you giving, right? You are giving 1, 2, 3, 4. At what rate are you giving this pulse? So that is what is called as the titanizing frequency, right? It is called as the titanizing frequency. The rate at which you are giving the impulse so that there is sustained contraction, right? So titanizing frequency, obviously, it would be just the reverse of contraction period, right? It would be directly, because more and more titanizing frequency, less and less would be the contraction period, right? And finally, at the exact full titanizing frequency, so there is a no point of any relaxation, right? It would be completely flat. Acha. The poison, the thing what is called as the Clostridium titani, right? It uses this frequency so that there is in this, when this infection goes, right, there is complete titany, and so much so that the body, it becomes like a bow, right? So body becomes like a bow. This is the bed, that's the nose, right? So body is like a bow, bends and contracts like a bow, right? Due to this frequency. So this was about the muscle part, right? Skeletal muscle. From tomorrow, we'll be starting a completely new. So let me ask you, what do you want? Say you want to start with cardiac, or you want to start with neural or renal. So you want to start with heart, brain, or kidneys. What would you like to start? Because then accordingly we'll take I'll arrange all the topics. Say respiratory system, cardiac. Okay. Say respiratory systems, even they are very important. And yes, I'll also take the X-ray chest, right, in this week only. So one day, right, I'll be giving to X-ray chest. So this will be beginning of the radiology, right? And 1 or 2 days will be devoting it for that. And it's good. So if we are learning cardiac, so then X-ray chest would be of much helpful, because you'll be able to see that what are the changes coming into cardiac conditions, right? So I'll keep on parallelly showing you the X-rays. Yeah, we can do that. Okay, so that's it for today, and we'll meet tomorrow the same time, and tomorrow we'll be starting with the heart. Okay, the cardiac one. Dystrophin? Sure. See, they are like supporting proteins. Yeah, dystrophin, right? Dystrophin is a supporting protein, right? You remember this, right? All those fibers, muscle fibers, right? All those filaments when they are arranged. So these are acting like a motors, but rest of the protein, they are for the support, right? So when there is dystrophin, this is just, you can say that it is like a brickwork. So when this dystrophin is mutated, so this, it leads to what is called as Duchenne muscular dystrophy. So you'll find that such patients, they'll be just collapsing. They can't even walk properly, right? As such, we'll be taking this Duchenne muscular dystrophy in more detail when we'll be proceeding further for the physiology. Acha, one more question which I wanted to ask you. What is going on in university lectures? What topics are going on? What exactly are you studying? So accordingly, if there is any overlap, I can cover those topics also. Because see, there is no point I get few emails about the various dissatisfaction that PowerPoint is shown, etc., etc. So just let me know the topics. What is going on, right? So instead of complaining, let's figure out a solution. So what topics are in progress currently? Or you can send a mail. Yes, go ahead, go ahead. Just do write your question. Muscle atrophy when there is decrease in the size. Dystrophy is when its function is lost, right? Atrophy, when the, in simple words, muscular wasting, muscle wasting. Dystrophy is when it is dysfunctional, when it is not working properly. Yeah, anyone, the topics which, anyway, you can just send me a mail, right, about the topics which are going on into your universities. So if there is any possibility, then we'll cover up accordingly. Okay, so thank you so much. And tomorrow we'll start with the cardiology, right? The basic physiology of heart. So starting from cardiac cycle to all the way, we'll learn about the ECG and also the radiological aspects, and we'll see the CT, MRI, right? And so much so that even some of the cardiomyopathies, even we'll be learning about auscultation, how to use your stethoscope, right? We'll actually hear those heart sounds and how to decipher it, right? So that would be for our cardiology, right? Which would be a combination of, yes, physiology, medicine, as always. Thank you so much and see you tomorrow. Bye bye.